This video is for days 8 and 9 of the short read sequencing analysis workshop. This is video 6, Isoform inference and abundance estimation with Stringtie. The goals for this video are to cover the basic operation of Stringtie for inferring gene isoforms and calculating their abundance. Also to describe when best to use Stringtie. The Stringtie ball-gown pipeline is an alternative to the feature counts DE-seq pipeline discussed in the previous videos. This pipeline takes mapped reads for some number of samples, along with an optional reference annotation, and determines what transcripts are present and which are differentially expressed. Stringtie is the tool that identifies and labels gene isoforms present in your data and then estimates their abundance. Ball-gown is the tool that compares abundance between your samples and determines which transcripts are significantly differentially expressed. We'll discuss ball-gown in detail in the next video. Stringtie is a command line tool which can either be run in a bash terminal or submitted as a shell script. Stringtie assembles transcript fragments using the spliced mapped reads and infers isoforms for each sample. It can also leverage annotation files to more confidently infer these isoforms. Using a splice graph model, it calculates abundances by calculating maximum flow through the network, removes that isoform, and then recalculates maximum flow for the next most represented isoform. Its output is any inferred annotation along with abundance, calculated in RPKM and raw counts. Stringtie is more effective at detecting isoforms with paired end data, as it uses the known orientation of the read pairs instead of just relying on individually spliced reads. In order to perform differential expression analysis, one must have a common transcriptome annotation to compare different samples. Stringtie can be used to merge the inferred transcript annotations generated for each of the individual replicates into a single common annotation. This common annotation can then be fed back into Stringtie to calculate abundance for each sample across the common set of transcripts. So, how do we run Stringtie? There are three main steps to running Stringtie. Each step calls Stringtie in a different mode. First, Stringtie generates a transcript annotation file for each sample individually. Second, Stringtie merges together the annotation files into one common annotation file. And finally, Stringtie uses the merge annotation file to recalculate abundance for each of your samples and formats the abundance data for input into ball-gown downstream. Each of these steps consists of a call to Stringtie, but in a different format. For step 1, Stringtie is called with the following options, at minimum. A single input BAM file. The G option indicates that Stringtie will use the reference annotation file to guide the assembly process. Output will include both reference and novel assembled transcripts. The O option indicates the output GTF file to contain the assembled transcripts. The P option indicates the number of processing threads to use. Stringtie will be run in this fashion on all samples before proceeding on to step 2. For step 2, Stringtie is called without an input BAM file, and instead it takes the list of GTF files generated in the previous step, one for each sample, and generates a unified, non-redundant set of isoforms, which are then saved to GTF files specified by the O option. The -merge option is necessary for Stringtie to run in this mode. The GTF list input should be a list of space-separated strings that are the full path to each of the individual GTF files. In merge mode, Stringtie can still use the original reference GTF specified with the capital G option to guide the merging process. Finally, Stringtie is again called to calculate abundance individually for each sample BAM file, but this time over the merged annotation file. At this stage, Stringtie should output the abundance estimations in a format that's suitable for input into ball-gown, the downstream differential expression tool. Using the -B option enables this output format. The "-E option indicates that Stringtie should only estimate the abundance of a given reference transcript present in the GTF specified by the "-G option. This means Stringtie will not, at this stage, attempt to construct additional de novo transcripts. Finally, the "-G option now specifies the newly merged annotation file in place of the original reference GTF. This completes the Stringtie process. For each of these 3 steps, there are many more options that can tailor the Stringtie output. Here we've only discussed the minimum requirements. So when is it appropriate to use Stringtie? Stringtie is an appropriate tool if you are studying differential splicing, if you anticipate your samples have unannotated transcripts that you would like to detect. It is more useful when you have paired end data. However, Stringtie is not necessary for nascent data, chip data, or any other type of sequencing data that's not going to be sensitive to splicing. If you are only interested in gene-level summarizing, the feature counts DE-seq pipeline would be more appropriate as it requires fewer total reads per sample to detect a sufficient signal, and the feature boundaries will be better defined. In the subsequent videos, we will discuss how ball gown utilizes the output of Stringtie.